This part of the text talks about how you map the genome. Now, you've done this before. If not in the lab, you've done this theoretically. You know, when you do crosses, you're normally given questions, and then you calculate, and then you given crosses, and then the observation on the progenies on whether they follow Mendelian or not, right? If they don't follow Mendelian, then what do you do? Then you tend to conclude that there's a recombinant, there's a recombination event that had occurred, and what you do then is to calculate the distance, correct? And how do you calculate the distance is basically looking at the number of recombinants over the number of progenies. And then what you get is the distance between two genes. And essentially, that is what you do with human genome mapping. Unfortunately, of course, you cannot do crosses for humans. So what you do then is to study natural crosses. And what I mean by natural crosses is looking or studying the pedigree. They actually establish families large enough so that you can do the calculation. You know, so for it's the same thing. You know, you look at a phenotype that is being passed on, and then two phenotypes, and then you see whether this phenotype is segregating together. Do they follow Mendelian ratios, or if not, you know, then probably you can see the distance between these two different characters. Now, most of these characters at that time were disease phenotype, but after a while, you know, there's not. I mean, you're talking about 20-30,000 of the human genome, and then, you know, you only know at that time about only about 1,000 different phenotypes. So one of the breakthroughs in genome mapping is the realization that you don't need to look at phenotypes. What you need to look at are polymorphisms in the DNA. What do I mean by polymorphisms in the DNA? Again, I'll give you an analogy. You are lost in the north-south highway. You know, I don't think you'll be lost, but let's say your car broke down in the north-south highway, and both at your sides are palm trees, oil palm trees, and then there's no signboards, but there is a telephone, right? You know, normally you see this SOS telephone, and then you call them up, and then they will ask you where you are. How do you tell them? How do you relay information of your position? Basically, if you look at the sides of the highway, there are these numbers, right? These are numbers indicating the position of where you are along the highway, and these numbers are meant to be unique. That means to say, if you are at position 232.4, just for argument's sake, along the highway there will be only one position that's 232.4, so they immediately know where you are. So the first idea is to have this kind of landmark along the human chromosome, so that if you establish landmarks, recognizable areas throughout the human genome, then when you find a piece of DNA, and you do some analysis on the DNA, you get some telltale signs, and then you know, oh, this particular piece of fragment contains landmarks that belong to chromosome 10, for example. All right, so that's the whole idea. And doing these landmarks, and well, they don't call them landmarks, they call them markers. And essentially, one of the first goals of the Human Genome Project is to establish this type of markers. And there are several versions of the human genome mapping markers. There are, initially they start off with, I think, about 9 million, one marker every 9 million, and then the target was one marker every 100,000. But as Human Genome Project goes on and on, you know, progresses, I think towards the end they got markers as close as one marker every 20 kilobases. And these markers come in the form of unique sequences along the chromosomes. They are called sequence tag sites, STSs. if the unique sequences represent expressed sequence, and they will be called express sequence tags. Express sequence tags may also refer to unique sequences that is found in a particular tissue type. For example, this gene is found, this EST is found in the retinal library, so you know that this gene is expressed in the retina and so on. All right. Take home message: STS sequence tag sites are markers and landmarks for the human genome. Okay, on to the next video.